morning, good afternoon, students. Welcome back to biology class. Last class we have started with genetic code, right? So in central dogma, DNA synthesizes the RNA, which process is called transcription. mRNA is carrying some genetic information which are present in the form of triplet codon, right? Whatever nucleotides they are arranged in three nucleotides will form the triplet codon. So now the genetic transcription, the information is going to be transmitted into protein synthesis in the form of a codon, right? So here in the last class we have seen genetic code. So genetic code which are present, properties or salient features of genetic code are very important to question there. Okay. Now, according to this genetic code is properties, it is universal. Okay, 61 codons are coding for different amino acids, specific amino acids. Okay, 3 codons are considered to be as a that is the nonsense codon. Okay, antisense codon, nonsense codons are present. And UAA, UGA, UAG. So these are the 3 codons which are stop codon. Okay, which will stop. And next character they are degenerate, and some of these triplet codon genetic code are the commonless, contagious. They are read continuously without any punctuation. Okay, and also these are collinearity. These are different universal and also amino acids. Least we have made 20 amino acid. Least also we have already seen. Now we are going to start a very important, again one more very important topic. Usually already we have studied in principles and variations also, and here also you are going to study about the mutations, types of mutation. Already you have knowledge about this mutation. Mutation is nothing but what? Sudden change in the DNA material. Sudden change in the DNA material, which we will call it as mutations. Right? So here mutations, the DNA material, always if the mutation takes place in DNA, variation takes place in mRNA. When the mutations variation takes place in mRNA, again the amino acid synthesis also changes. When amino acid synthesis changes, again the protein synthesis also will become abnormal. The protein synthesis means one will leads to the problem to the another thing. Okay. Now sudden change. This was read by this material or the mutation was explained, as you know, it was explained by the Hugo D. Varies. You know that is Hugo D. Varies. He was the first person to discover about the mutation. So, especially he has observed the mutation in evening prime rose. Evening prime rose. The prime rose, which is named as Oenthera lamarckiana. Okay, the prime rose. Oenthera lamarckiana. Okay, Oenthera lamarckiana is the scientific name of the prime rose. First, he has discovered the mutation in the what Oenthera lamarckiana, and he also told that is mutation, the components. Okay, the products which causes the mutation, they are considered to be as mutagens. Mutagens may be biological mutation, chemical mutations, or physical mutations, or can also be Means mutations may be either through the physical or through chemical, or mutation also takes place because of virus, bacteria. So biological mutations are present. Okay. So the mutation, different types, agents, this gene, they are, so overall mutation, chromosomal mutation is also there, and gene mutation is also. Mutation are of two types, that is, okay, chromosomal mutation, we have, and gene mutation also we have. So chromosomal mutation we are not going to study in this chapter because already we have completed different types of chromosomal mutation in principles and variations, right? In this class we are going to see different types of gene mutation. Gene mutation was first discovered by Muller. Okay, Muller explained about mutations also. Okay, he was gene mutation. The mutation may also take place in the genes, which was given by the Muller. And the mutations term and also discovered mutations are because of the chemical mutation, and biological mutations are also present, which causes the different types of mutation by mutagens. Mutagens means what? The agents which causes a mutation, that agents are called mutation. That agents are called mutation. Try to understand. Mutation means sudden change in the DNA sequence. But mutagens are nothing but the agents which causes mutation. That agents are called mutagens. Understood? Next, coming to the flowchart of the gene mutation. Let us see the gene mutations. We are concentrating on gene mutation, which are divided into base substitution, okay, and the other side is a frame shift mutation. Frame shift mutation. The base substitution, again, it has different types. Base substitution, they are, and also inversion we have. So transition, transversion, inversion. We have transition, transversion, inversion. Okay, three types: transition, transversion, and inversion. Frame shift mutation. In frame shift mutation, okay, we will see later. First, let us see the base substitution. Okay, gene mutations are for base substitution, frame shift mutation. Base substitution are of three types. Let us understand what do you mean by gene mutation. So here, sudden change in the nucleotide will result in the gene mutation. We are talking about the particular only one nucleotide. Okay, either it may add, delete, and all. Okay, if you see the number of the base substitution, if you take, the number of nucleotides will always remain same. Some variation, adding, deleting is taking place. But in case of substitution, the number of nucleotides, remain same. Either it is a transition, transversion, inversion, base substitution is nothing but something nucleotide is adding or deleting. So there the number of what nucleotides are same. There is no decrease and there is no increase. Some mutation is taking place in the nucleotide, but even the the number of nucleotides remains same. Remember that while studying the example, you will be coming to know about the base substitution. In base substitution, the number of nucleotides, either they will be adding or they will be deleting the nucleotides. But even after that, some changes is taking place. But even after that, the nucleotide number of nucleotides will remain same. There is no increase, there is no decrease. Okay, so this type of mutation is substitution. After this, the base substitution definition, if you see, so the DNA mutation occurs any of the nucleotides. So the definition of base substitution definition. So in the base substitution, the mutation occurs here. The mutation occurs in DNA. Okay, the mutation is taking place in the DNA of the nucleotide, or you can call it as a nitrogen base nucleotide. The mutation taking place in the DNA of nucleotide or nitrogen base, any of the nitrogen base is any of nucleotide or nitrogen base is substituted by another nitrogen base, substituted by another nitrogen base. So the number of nucleotides, why the reason is, why the number of mutation is taking place even though the number of nucleotides are remaining same. Why? The mutation is taking place in the DNA. DNA are made up of all nitrogen base, the nucleotides. So one of the nucleotides are replaced or substituted by another nitrogen base. Okay, we will see what is. This is the definition of the base substitution. What do you mean by that? The mutation is occurring in the DNA. So DNA of any nucleotide, okay, any nitrogen base is substituted. Okay, it is substituted by another nitrogen base. Okay, so one nitrogen is taken and another nitrogen base is substituted. So any one nitrogen base is added. So in this case, it is called base substitution. Understood? What happened? The number of nucleotides will remain same, but one nucleotide is removed and it is replaced or substituted by the another nitrogen base. That is called base substitution. Okay. So now let us see what is transition. We are going to apply for transition now. Base substitution. First one, let us understand. Okay, one nucleotide is taken and another nucleotide is substituting here. In these three, the number of nucleotides will remain same, but the variations one is replaced by the another. Let us see transition. Okay, transition, base shift, base substitution. So in case of this, the purines are here, the purines are replaced by another purines. Okay, purines are replaced by, for example, what are purines? Purines may be replaced by the another purine. Okay, so purines are replaced by the another purine, and again this G may be replaced by the A. What is transition? The replacement of one purine by another purine. In case of, if by on the DNA, it should be A, T, C, G, right? In case of A, it is G. In case of G, it may be A. So purines, if they are replaced by the purines, then this type of mutation we will call it as transition. Are you understanding this? Try to understand. The purines are replaced by the another purine. A, in place of A, G will be present. In place of G, A will be present. So this is also mutation only. Okay, this is also mutation. Or else sometimes pyrimidine may also be replaced by the pyrimidine. In a transition, sometimes pyrimidine is also replaced by pyrimidine. In case of transition, one more possibility is that. What is that? Pyrimidine can also be replaced by the pyrimidine. Okay, so the possibility is there. The pyrimidine is also replaced by the pyrimidine. For example, pyrimidine means instead of T, there is C. Okay, instead of C, there will be T. Understood? Instead of, so this is either the transition means what? On DNA, these are nucleotides. Okay, nucleic acid molecules are nucleotides. The purines are replaced by purines. Pyrimidines are replaced by the pyrimidines. So either purines are replaced by purines only, pyrimidines are replaced by pyrimidines only. So this is called as what? Transition. What is called as transition. So sometimes purines. Understood? Next, let us go for the another transversion. Understood transition? Transition means what? The mutation is taking place by replacement of one purine by the another purine. So either in place A should be there, in place of A G is there, in place of G A should be there. So there will be what? One purine is replaced by another purine, or one pyrimidine is replaced by another pyrimidine. Let us see in the transversion. Transversion. In case of transversion, when purines are replaced by pyrimidine. Here pyrimidine only. But there is a possibility, purines are replaced by the pyrimidines. Okay, purines are replaced by the pyrimidines. there is a possible now. How? Okay, purines you can take A or G can be replaced by C or T. Okay, purines and pyrimidine can be replaced. For example, one simple flowchart you can see pyrimidines can also be replaced. Pyrimidines can also be replaced. Not only purines can be replaced by pyrimidine. Pyrimidine may also be replaced by the purine. C or T may be replaced as A or it may be replaced by the G. Understood what is transition? Purine is replaced by purine. Pyrimidine are replaced by pyrimidine. These are the one type of mutation. In case of transversion, purines in place of a DNA segment, instead of purine, there may be pyrimidine. In place it should be pyrimidine, but in case of pyrimidine, purines also will be replaced. Okay, so this is called the transversion. Okay, what it is called? Transversion. Purines are replaced by pyrimidine. Pyrimidines are also replaced by the purines. Understood? So this is called the transversion. Next. So transversion and inversion, we can give example here. That is, so this question was asked. Okay, remember? And if you take here, okay. Only thing to make you to clarify transition and transversion. So here we are going to see transition and transversion. That is A and G. A and G are what? Purines. Okay. A can be replaced and simultaneously G can be replaced. Right? Okay, this is called as transition. What it is called? A in place of A, G can be replaced. In place of G, A can be replaced. Simultaneously, in place of T, C can be replaced. And in case of T, purines or pyrimidine. This is called transition. Right? A can be replaced. Purines are purines and pyrimidines are pyrimidines. Now, in case of transversion, A to, so this A to D, T may give rise to G, G okay, this is transversion. In case of transversion, so these are pyrimidines. What are the pyrimidines here? C, T are pyrimidines, A, G are purines. Correct? These are purines. These are pyrimidines. Clear. Transition means what? Purines. Purines are replaced by purines. Pyrimidines are replaced by pyrimidine. This is called transition. But according to transversion, purines can be replaced by the pyrimidines. Again, purines can be replaced by the pyrimidines. both reaction we are showing, and pyrimidines are replaced. So these are called transversion. Okay, all these sites are called as what? Transversion. Middle, purines are replaced by purines. Pyrimidines are replaced by pyrimidine. This is called transition. These are called, this is 1, this is 2, this is also 3. All the 4 are transversion. All the 4 are transversion. Now, understood? Let us go for next one, that is inversion. Understood what is transition? Purines are replaced by purines. Pyrimidines may also be replaced by pyrimidines, that is called transition. In case of transversion, purines are replaced by the pyrimidines. Okay? Or pyrimidines may also be replaced by the purine. Next, third one is inversion. Okay, inversion means what? Inversion means if the DNA, see this is a DNA 5 prime, 3 prime direction. Okay, I am taking A, B, G, C, T, G, A, T, T. The inversion will occur. Inversion means reverse. Okay, so there is no deletion, there is no addition, just there is a change in their position, but number of nucleotides remain same. It is same in transition, it is same in the only replacing, but no deleting, no adding. Okay, the number of, already we have written, the number of nucleotides will remain same. So by reversing, what happens? So take, this is a DNA. So G T A. Ulta ho G T A. And these will be same. T G A T T will be same after inversion. Okay. So DNA structure. Inversion means reverse. The nucleotide will be reversed. This type of mutation is called as inversion. Inversion means reverse. Inversion means what? Reverse. Understood? So, 3 different, what we have studied in gene mutation, base substitution, frame shift mutation we will study next. In base substitution means what? The mutation takes place in the DNA of the nucleotide, okay, of the single nucleotide is replaced by the another nucleotide. One nucleotide is replaced by the another nucleotide, like purine is replaced by purine, like pyrimidine is replaced by pyrimidine. If same type are replaced, then it is called transition. The number remains same. Okay, transversion. Purines are replaced by pyrimidine. Pyrimidines are replaced by purine. So this type of mutation is called transversion mutation. In case of inversion, the triplet codon, the codon will be reversed. Okay, if the mutation takes place in ATG, the inversion they will become invert, they will become reverse. Okay, that is called inversion. Understood? That is called as inversion. And next example for base substitution example, we can give sickle cell anemia. Okay, we can give sickle cell anemia as an example for the base substitution. What is sickle cell anemia? We have already studied in principles of inheritance, right? So there what happened? Glutamic acid, that is in the hemoglobin, that is beta chain, 6th position. Hemoglobin have alpha beta chain. Beta chain, in beta chain, 6th position, glutamic acid was replaced by valine, right? So what happened there? One amino acid, instead of one amino acid, another amino acid is replaced. That is called sickle cell anemia. Understood? Which comes under base substitution. Which comes under base substitution. Next. After this, next we will go for the frame shift mutation. Let us go for, you can see here, purines may A can be replaced by G, so one line. And also G can be replaced by A, another line. Simultaneously T can be replaced by C, one line. And simultaneously C can be replaced by T, another line. So this plus mark indicates transition. Purine should be replaced by purine, pyrimidine should be replaced by pyrimidine. But in case of transversion, what happens? A can be replaced by C, C can be replaced by A. C can be replaced by G, G can be replaced by C. G can be replaced by T, T can be replaced by G. So this outer layer is called transversion. Let us enter into the frame shift mutation. In case of frame shift mutation, it is of two types. Backward frame shift mutation. Okay. One more. Forward frame shift mutation. So let us understand what is this frame shift mutation. Okay, so we have completed base substitution, number of nucleotides was remaining same. But in case of frame shift mutation, either deletion or addition of nucleotides will take place. Either deletion or it may be addition of nucleotides. With example, we are going to see, and very important question this one. Okay, we have frame shift mutation, point mutations also we have. So frame shift mutation, either deletion or addition of, understood? So either deletion or addition of the nucleotides. So here, so frame shift mutation, either deletion or addition. If any one nucleotide delete or in case of frame shift mutation, what happens? Deletion or addition of nucleotide will take place. Where it will delete or where it will added, from that point onwards on a DNA, from that point the segment of the DNA sequence change. But here, in case of this, what happened? The number of nucleotides was remaining the same. Right? The number of nucleotides was remaining same. Understood? But here in frame shift mutation, what is going on? In frame shift mutation, either adding or removing the nucleotide from either adding or deleting, from that point onwards, the DNA segment, the DNA sequence changes. Okay, the DNA sequence will be changed. For example, so frame shift mutation, frame shift mutation. So we are taking the DNA segment. Okay, we are taking the DNA segment that is from the 5 prime to the 3 prime. Okay, the 3 prime, that is ATGG, right? And they are always present in the triplets. The codons are present in the triplets, correct? Now, according to frame shift mutation, what happens if in case of this A is removed? Okay, in case of A is, we are going to change there. A is removed. In this condition, what happens if one nucleotide deleted or added? So then the sequence will be changed. For example, I'm going to add or remove this A. What happens? I'm going to remove A. The reading of the sequence of the DNA will become same. Up to here it is same. What did I told in frame shift mutation? From where adding or deleting of the nucleotides, from there onwards the sequence of DNA changes. From here onwards it will change, so I will keep as it is. Okay, G and here A is removed. Then what happens? We will can take here G T A C G. See here, one codon is decreased. Okay, because of the removal of the one nucleotide, either it may decrease. If we go on adding one more A, again sequence will be changed. Okay, we will see some another examples also. Now, so what happens if one nucleotide is added or deleted? From there, the DNA sequence changes. So you can see from here the DNA sequence change, up to here the sequence is same. There is no change at all. Here we have removed, we have deleted, we are going to add. From that point onwards, there is a slight changes can be seen. Let us see other examples of frame shift mutation. So entire, that is sequence, in NCRT also they have used one of the example RAM and all. So that we will give later. Another examples we can see. One example I am going to give you, that is Tom, Big Cat, A Cat. Example. We are using the term which is having only 3 words, 3 alphabets. Tom, Big Cat, 8, rat. This is a thing, this is a DNA sequence now. Having three words we have selected now. Now what happens according to frame shift? So what is this? This is a first codon, this is a second codon, and this is a third codon, and this is what fourth codon, and this is fifth codon. This is a normal condition. Now what I will do is, I am going to add, okay, at here, I am going to add a R. What I am doing now, I am adding here, this sequence, adjacent side, I am going to add a R. So, from where the point up to here it is same. Cat, up to cat it is same. So now what it will become? It will be same. Tom is same. There is because we didn't have anything here. Big is also same. Okay, cat is also same. But now here we are adding what? We are adding AR. AR and A. AR A. Now AR A. TER. TER. Okay, then we are left with A T. A T. Okay, so sequence has changed, right? Tom is same, big is same, cat is same. But what happens here? The sequence of DNA changed. The sequence of DNA has changed in the nucleotide. Is called frame shift mutation. Frame shift means what? Either it may be, so this shifting is of two types, forward frame shifting or backward frame shifting. Either forward or it may be the backward. Let us understand this. Okay, shifting is taking place where we will add the nucleotide from that position onwards. The sequence of DNA will be changed. The sequence of DNA will be changed. Understood? Let us understand two different types of frame shift mutation. First, now next, forward and backward. So here, let us understand forward frame shift mutation. So forward means we are inserting or addition. Inserting means what? Addition of nucleotides. What we are doing here? Forward means addition, backward means deletion. Okay, what do you mean by forward frame shift mutation? There is an addition or insertion of nucleotides. Some nucleotides we are adding, or if we are adding here, either adding or deleting. If we are adding, it is called forward. If we are deleting, it is called backward. Understood? Now, forward frame shift mutation, we are inserting or addition of the nucleotides. Let us understand the example. So, example if you take, so example for the forward thing, A, U, G, okay, G, U, A, U, C. This is a normal. Now what we are doing is, here one A is added. Again the sequence, what happens here? We are adding this. So here it is same. A, U, G. Up to A, U, G it is same. So the change is taking place here. A, G, U. The C will shift here. C, A. C, A, U. Okay, and one C is left here. Forward, what happened? One C has shifted forward. So this is called forward sequence. Forward sequence. The sequence changed here. This was normal. This is forward frame shift mutation. Sequence change here. Whereas the next backward shift example. So okay. So forward shift mutations we can give example for frame shift. Example is muscular dystrophy, muscular dystrophy. Backward, here insertion, addition of nucleotides, here deletion of nucleotides. Same, we can see here in backward AUG, here we can remove. Let us remove this U. U is removed. What happens to the sequence? This will be say U G G C C because U is removed. G C C A C A and U C. This is called backward mutation. Forward mutation we were adding. Backward mutation we have removed one, so two are left, one is missing here. So backward shift is taking place. Okay, forward as well as the backward. So some NCERT example. Let us take in backward. We can take NCERT examples now, which is given in your textbook. So backward. NCERT example, they have given Ram has cap. Ram has cap. Okay, Ram has cap. Now what we will do for this line, R is inserted. R is inserted. What happens here? In backward, so forward, after inserting, Ram has cap. Okay, here we are going to insert R here. Correct? Up to here it will be same. Ram has. Here what happens? It is the sequence are going to change. R, C, A, then P. So this type is called as what? What happened? The sequence changes. Again here we are going to add E is inserted. This is a forward. Same. Here the sequence will be what? The sequence will be same. Here we are introducing RE. Okay. Again, Ram has, okay, REC. This will shift now. Okay. A, P. Okay. Now we will, after RE, we will insert D. D will be inserted. What it will become? This sequence has changed, right? Now D should be added. There is same here, Ram has. Now we have to add R, E, D will be added here. Here E was added, here D is added, red, C will shift here, cap. This is forward mutation. Forward frame shift mutation. So what examples we are taking here? The examples which we are taking here are forward and backward. Backward means we are deleting the nucleotide, but in forward we are adding or insertion of the nucleotide. You can see Ram has cap. I am introducing R here, RCA. This will shift forward. To after R, I am introducing E. It will become REC, A shift here, AP. Where from here almost everything the DNA sequence is same. From here onward the DNA sequence is changing and it is forwarding and it is increasing. Okay. So RD I have also. Ram has red cap. The sequence has completely changed. If I remove this, again it will become normal. Ram has cap. First Ram has cap we have taken. Red codon has been introduced. Okay. Next. So next class we will discuss about the point mutations. Okay. So clarify. Gene mutation are of two types: base substitution, frame shift mutation. Base substitution, number of nucleotides remains same. So here what is going on? Number of nucleotides increased. Okay, or it is decreased in this case, in this frame shape. But here we were adding, we were deleting. Transition, purines are replaced by purines. Pyrimidines are replaced by pyrimidines. In transversion, purines was replaced by pyrimidines. Pyrimidines was replaced by purines. So number was same there. There was no change. But here backward means there was decreasing, forward means because of mutation, number of nucleotides we were adding. So the sequence, the number of nucleotides was changed. That is called frame shift mutation. Example here, muscular dystrophy. For base substitution, sickle cell anemia we can take example. Thank you students. We will see point mutation is